So I would like to introduce the second presentation from the team working on the massive parallel sequencing of the plasma nucleic acid for the molecular diagnostic of cancers. The project coordinator is Professor Dennis Lowell, and today Professor Alan Chen will present the finding on this film-based project.
Thank you, KW. It is exciting to be here. So, as a young member of the team, I'm not going to present the update of the progress of this project, but instead, I'm going to share with you the excitement and joy of doing scientific research as a young investigator. So, a lot of the young people are interested in science. But however, in Hong Kong, I think only very few of them would commit to a career of scientific research. I think the reason can be complex, but some practical reasons may be of concerns to them and to myself. So one reason is the lack of job security in scientific research. And others may be the inferior financial return for researchers compared with other professionals, including medical doctors, lawyers, and investment bankers. But I think we need the courage and passion to do scientific research here and to commit ourselves to the scientific research career. So I want to use my experience as an example to show you how interesting it is to do scientific research and in Hong Kong. So when I graduated from the medical school and received training, specialist training in gastroenterology, I decided to go back to school to pursue my PhD studies because of one reason, because of one interesting finding made by this young gentleman in the picture. He is Professor Dennis Lowell, and in 1997, he discovered that when a woman is pregnant, the DNA would be released from the baby into the mother's circulation. And I find this discovery very interesting and is a conceptual breakthrough. Because in the past, we think that it was generally believed that the circulations of the baby and the mother is completely separated to avoid adverse immunological reactions, which may damage the baby. But he found that when a woman is pregnant, the baby's DNA can be released by the baby into the maternal circulation. And I think there would be a lot of practical use of this new phenomenon. And for the benefits of those who are not working in life science, I will briefly introduce what is DNA. DNA is the genetic material governing how we look like. So normally, DNA is present in the nucleus of cells and packaged in a larger unit called chromosomes. But if we take a blood sample from our circulations and after centrifugation to separate the blood cells and the liquid portion of the blood, which is called plasma, we can detect a very low concentration of DNA in the blood plasma. And this DNA is released from different cells over the body. So it is a rubbish bin collecting the unused materials from the cells. But because of this, it is a treasure trove to do molecular diagnostics because it receives all the information from different parts of the body. So especially the phenomenon of having fetal DNA in maternal plasma can be used for prenatal testing purpose. In the past, if we want to obtain genetic materials from an unborn fetus, we need to stick a long needle, like this long, into the tummy of a pregnant woman. And this procedure is very scary and also carry risk of miscarriage. So the presence of fetal DNA in maternal plasma opens a window so that we can obtain the fetal materials non-invasively by just taking a tube of blood from the pregnant woman. So the most important thing is to crack Down syndrome because to detect Down syndrome is the most common reason for a pregnant woman to carry out prenatal testing. And for Down syndrome, it is caused by the presence of an extra copy of chromosome 21 in the genome. So this extra copy would cause some adverse events in terms of intelligence and behavior. So, but to detect Down syndrome non-invasively by plasma DNA analysis is technically challenging. The reason is in a pregnant woman, only around 10% of the DNA comes from the baby, and the remaining 90% of the DNA actually comes from the mother herself. And these DNA are chopped into very small fragments, and we cannot differentiate which one is from the baby and which ones are from the mother. So it took us around 10 years to solve this problem. And during the 10 years, there are two major developments that provide us the necessary tools to solve this problem. The first advancement is the finish of the Human Genome Project. Human Genome Project is a concerted effort all over the world. So scientists all over the world work together and spent 13 years to come up with the draft of the reference human genome. It consists of around 3 billion nucleotides and approximately 20,000 genes. The cost of this project is $3 billion US. And other advancements is the development of the second generation of sequencing machines. So using massively parallel sequencing, we can sequence hundreds of millions of DNA molecules simultaneously, so that we put in a chip, then we have the readout of these nucleotide fragments. So, and this, with the latest models of this machine, in 3 days, we can sequence the human genome 300 times. So, in the first draft of human genome, we spent 13 years to come up with one human genome. But now, in 3 days, we sequence it in 300 times. So we are now 100 million-fold faster than the previous version. So our solution to solve the prenatal diagnosis is that we take a tube of blood from a pregnant woman and separate the blood cells from the plasma. In the plasma, we sequence all the DNA fragments, and it includes DNA from the mother and the baby. So in this diagram above, the red ones are the DNA from the babies, and the black ones are the DNA from the mother. We don't need to separate them. We just need to sequence everything in the plasma samples. And after the sequencing, we would align these DNA fragments to the reference human genome. The purpose of this is to determine the chromosomal origin of each DNA fragments. And after that, we count the number of fragments originating from every chromosome. And because we have counted millions of DNA fragments, we can calculate the chromosome dosage very accurately in the plasma DNA. And this, because of the presence of an extra copy of chromosome 21 in the fetus for those affected by Down syndrome, then we would expect an increased dosage of chromosome 21 in those women carrying a Down syndrome fetus. And this method is extremely accurate. The diagnostic accuracy is 99.7%. It is more accurate than any clinical laboratory test that we used routinely. So, after the success of cracking the Down syndrome, we think that this methodology can also be applied to the detection of cancers, because the presence of a cancer in the body is very similar to the presence of a baby in the pregnant woman. And the placenta is as invasive as the tumor to the uterus. So we think that we can modify the technology to detect cancers. And one objective is to develop a generic test for the detecting all types of cancer. I think this is possible because copy number aberrations is very common in all types of cancers, and virtually we can detect copy number aberrations in all types of cancers. So this is the solution that we come up with. And in this circles plot, the chromosomes are arranged in a clockwise manner, so that in the outer circle, you can see the chromosome 1 to chromosome 22 arranged in a clockwise manner. And then we first analyze the tumor tissues from a liver cancer patient so that you can see many dots here. And each dot represents a 1 megabase region. So there are 3,000 dots in this chart. So the gray dots represents that the region that did not have any copy number aberrations. And the green dots means that there is some extra copies of chromosome in this particular region. And the red dots mean there is some deletion of the chromosomal region. And first, we observed some copy number aberrations in the tumor tissues. And then, if we look at the plasma samples collected from the same patient, we can see very similar patterns when compared to the patterns of copy number aberrations that we see in the tumor tissues. So the plasma just reflects the information of the tumor. And then after the surgical resection of the tumor, all abnormalities are gone. So it proves that the signal that we detect in the plasma is actually coming from the tumor. For subjects with chronic hepatitis B but without cancers, then we can see that the results are very clean. There is no red and green signals here, so we don't see any abnormalities. And this method can be extended to the detection of all other types of cancers, including breast cancer, lung cancer, smooth muscle sarcoma, neuroendocrine cancers. Basically, all types of cancer you can name, we can detect the copy number aberrations from the plasma. But is it a good test? So now we have a test that can detect all types of cancer, but is it useful clinically? I think may not. Because imagine if a patient comes in and you test the blood and detected some abnormalities. And then what information you are going to provide to the patient. So I would say, okay, we detect something, you have a cancer, but it can be any types of cancer because we can detect anything. So we don't know what to do next. Either to have an MRI brain, CT lung, or CT abdomen, or something in the bone. So it's not very useful. So the next question is to identify the source of tumor-associated abnormalities. But it is very difficult to do because the DNA sequence from different cells are the same. So the DNA sequence cannot differentiate a brain cell from the liver cell from the lung cell. So what can we do? So we rely on another phenomenon called DNA methylation. DNA methylation is that there is certain parts of the genome, and there is a methyl group attached to the DNA. And this DNA methylation is to switch on and switch off a gene. Because if a gene promoter is not methylated, then the transcription factors can bind to the gene and begin and switch on the gene expression. Then the gene is switched on. However, if the gene promoter is methylated, then the transcription factor cannot bind to the gene and switch off. The gene would be switched off. And this mechanism is very important to control which genes are switched on and which genes are switched off in the cells. So it gives us an opportunity to design which DNA fragments, from which organ these circulating DNA fragments come from. It can be from the liver, from the lung, and from the blood cells. So if we look at the different tissues, they would have different methylation patterns because for different tissues and different cells, they need to switch on different sets of genes so that the methylation patterns on different tissues would be different. And based on the difference in DNA methylation, we can determine the tissue origin of the circulating DNA. And this, we can, using a genome-wide bisulfite sequencing to analyze the whole genome for methylation patterns and compare it with the tissue-specific methylation profiles. And we call this method a plasma DNA tissue mapping. We can determine the contributions of different tissues to the plasma DNA. And this method would be useful to determine where the abnormalities come from in the cancer patients. Because when there is copy number gain in the cancer cells, and if we look at the tissue contributions, then the regions with copy number gain, because of the extra copy of the chromosome, then the tissue would release more DNA into the circulation, giving rise to a higher contribution of this particular tissue. But in contrast, if a particular chromosomal region is deleted in the tumor cells, then because of the loss of a chromosome, then less DNA would be released by the tissue, by the tumor tissues into the circulation, giving rise a lower contribution in the blood plasma. And then we can calculate the difference in contributions in the regions with gains and loss, and the type of tissue corresponding to the copy number changes would give the maximum difference in the two parts. And then, if we look at the tissues without copy number aberrations, then we would expect a close to zero difference between the two types of contributions. So if we use these methods and look at liver cancer patients, then the liver gives the maximum signal. And for lung cancer, the lung contribution is light up, and the colon cancer patients, the colon is indicated as the potential source of the abnormalities. So then, if we have a method to detect all types of cancer and locate the cancer, what is clinical utility? Probably we can use it to screen for cancers in patients who are asymptomatic. But to do this, we need to answer one important biological question, is would a small tumor release sufficiently large amount of DNA into the plasma to allow detection? This question is very important if we want to use this method to screen for early cancers, because we know that the half-life of circulating DNA is only around 30 minutes in the circulation. So we need a large amount of DNA to be released from the tumor into the circulation to allow us to detect this. So to do this, we use the nasopharyngeal carcinoma as an example, because this tumor is very common in China, especially in the southern part of China, and Hong Kong is in the middle of the high incidence area. And the second reason is this cancer is closely associated with the Epstein-Barr virus infection, and we can detect the Epstein-Barr viral genome in basically all tumors of NPC in Hong Kong, so that we developed the Epstein-Barr virus DNA as a surrogate marker for NPC with a very good sensitivity and specificity. So we carry out a 20,000 subject NPC screening project over the last 4 years and completed this earlier this year. So we focus on subjects with a high risk of developing NPC. So we recruited subjects who are males, ethnic Chinese with age 40 to 62 years, because we want to see whether the screening can identify very early cancers. So we exclude any individuals with symptoms of NPC. So the study arrangement is that we take a tube of blood from the participants at the time of enrollment. If they are positive for the plasma EBV DNA, then we will repeat the analysis in 4 weeks' time with another sample. And if a subject is persistently positive for the plasma EBV DNA on these 2 occasions, we will carry out nasal endoscopy as well as MRI, and we will perform yearly follow-up, phone follow-up of all these individuals to follow up if they have developed NPC or other types of cancer. So we carried out 143 recruitment sessions over the last 4 years and recruited over 20,000 subjects. And all these recruitment sessions were carried out during weekends. So as a result, my golf handicap increased by 20 strokes over the last 4 years. And we found that 5.5% of them were positive for EBV DNA at baseline. And 4 weeks later, if we repeat, only 1.5% of them had persistently positive result. So we performed a nasal endoscopy on 300 subjects, and 275 of them also received MRI, and 25 had some contraindication to MRI. And 9 subjects, unfortunately, refused further investigation despite the persistently positive results. So we identified 34 cases of NPC. And before looking at them, I would introduce to you the statistics of NPC in Hong Kong. So in 2013, we have around 800 cases of NPC confirmed in Hong Kong, but only around 20% of them are of stage 1 and stage 2 disease, and majority of them had stage 3 and stage 4 disease. This is not ideal in terms of treatment because stage 1 patients and stage 2 patients had much better survival compared with those with stage 3 and 4 disease. So in our cohort, we identified 34 cases, and 16 of them, almost half of them, had stage 1 disease. And stage 1 and 2 patients add together account for around 70%. So there is a very significant improvement in the stage distributions. And as a result of this change in stage distribution, we demonstrate a superior survival for these patients. And for only one patient identified by the screening program developed disease recurrence, and the hazard ratio is 0.1. So it means that through screening, we reduce 90% of the chance of disease recurrence in this group of patients. And unfortunately, 9 subjects refused further follow-up, and 1 of them developed advanced NPC 32 months after the screening. And he died 2 months later. I strongly believe that if he received further investigations after a positive screening test, I think this tragedy can be avoided. So in addition to the exploration of the clinical applications, we are also interested in the biology of circulating DNA. One interesting question is, what is the size of DNA? And when Dennis asked me to do this project when I was the first year PhD student, and I tried to pull out the ruler to measure the DNA, but I find out the DNA is very short, and I cannot use a ruler. So how can we measure the size? So in one particular project, we constructed the whole fetal genome by just sequencing the mother's plasma DNA. So we proved that the whole fetal genome is present in the maternal plasma. And through a technology called paired end sequencing, we sequence both ends of a plasma DNA and then mapped to the reference human genome to determine the coordinates of the 2 outermost nucleotides. Then we can deduce the size of the DNA fragments. So this is the size profiles of DNA from the baby and from the mother in the maternal plasma. So you can see that the size profiles of the fetal DNA and the mother's DNA are a little bit different. And the fetal DNA represented by the blue line is on the left side of the red line, which represents the mother's DNA. It means that the fetal DNA is shorter than the mother's DNA. But in both types of DNA, we can observe a prominent peak at 166 base pair, but in the fetal DNA, there is another prominent peak at 143 to 146 base pair. So what is the biological reason leading to this observation? So we need to look at how DNA is packaged in the nucleus. The DNA is caught around some proteins called histone, and each histone complex, actually there is 1.6 rounds of DNA calling around it. And for the complex, it involves 146 base pair of DNA. And for successive histone complex, they are connected by a piece of DNA, which is called linker, and is 20 base pair. And add together is 166 base pair. So the 166 base pair peak is the core histone DNA together with the linker. And the fetal DNA lacks this linker. Probably it was chewed somehow when it is released into the circulation. And can we extend these observations to the plasma DNA profile in cancer patients? Yes, we use the liver cancer as a model to do this. So if we look at the size distribution of DNA in healthy subjects, we observe a very nice peak at 166 base pair. Then if we look at the liver cancer patients, then we would like to divide them according to how much tumor DNA is present in the plasma. So if they have very low concentration of tumor DNA in the plasma, we observe the blue lines are on the right side of the black line. It means that there is slightly prolongation of this DNA. But when the amount of tumor DNA increases progressively, then we can see the curves are moving towards the left side. It becomes shorter and shorter. So if they have more than 80% of the tumor DNA in the blood plasma, the plasma DNA becomes very short. So what is the explanation of this? We hypothesize that when there is a tumor in the liver, then the short DNA would be released from these tumor cells into the circulation. However, when there is very small tumor, the tumor would invade to the neighboring non-tumor regions, releasing relatively long DNA. But how can we prove this? So, as I have mentioned, the copy number aberrations would lead to an uneven release of DNA from the tumor cells. So for regions with amplifications of gain of copy numbers, then it is enriched for the tumor DNA. And for those with deleted regions, there is relatively depletion of the tumor DNA in the circulations. So if we compare the size distributions of these two regions, we can confirm that the DNA released from tumor is actually shorter, so that we can see that from this size distribution curve, we can see that the green regions is actually on the left side of the red region, meaning that they have relatively short fragment size for regions which are enriched for the tumor DNA. So I have shown you what we have done over the last 15 years. So what have we done? What benefits have we brought to the society? So one important benefit is we developed a lot of intellectual properties. We have filed hundreds of patent applications over the last 15 years, divided in more than 40 patent families. So these patents are very important for the development of the field because it can grant biotech companies the exclusive right to practice these methods, so that it is a very strong incentive for them to invest further in the field. So, and for the non-invasive prenatal testing, because of these patents we licensed to some international biotech companies, it results in millions of tests performed in 2016 in over 90 countries in the world. And it results in a 40% reduction in the number of invasive procedures related to prenatal testing. So it saves a lot of baby. And then we think about whether it is the best interest to Hong Kong if we license all our patents, valuable patents, to international biotech companies. We think it brings some interest or brings some benefits to Hong Kong, but it is not the maximum benefit. So to maximize the benefit to Hong Kong, the three of us, Dennis, Rosa, and myself, decided to found some biotech companies ourselves. So we developed we founded two biotech companies, and both are based in the Hong Kong Science Park. So the Exelon is focused in the development of non-invasive prenatal testing, and it has the largest market share of non-invasive prenatal testing in Hong Kong now. And Serena focused on the development of DNA-based technology for cancer screening. And interestingly, in the same time, other parts of the world realize that cancer screening using DNA technology is a very valuable business. So there is another company formed, and the name of this company is called Grail. And it is a subsidiary of the largest sequencing company in the world called Illumina. So Grail is a subsidiary of Illumina. And because of this, they think that probably it is worthwhile for us to work together. So there's a merger between Grail and Serena earlier this year. And the combined entity is now the most valuable biotech company in liquid biopsy in the world. So I think it gives us a lot of opportunities to move forward and the necessary resources to move forward. So in addition to the site in Science Park, we have also a new site in APM in Kwan-chee with a 12,000 square feet laboratory to provide service in the first half of 2018. So in addition to monetary return, we also focused on the education and training of people. So this is our group Proto. You can see that there are a lot of young faces here, but I want to highlight some of them. So you can see that this gentleman is called Jackie Lam, and he got straight 10 A's in the Hong Kong certificate examinations. He is a fully qualified ENT surgeon, but he decided to quit and join us to do PhD training. And Tim Zhang is a Rhodes Scholar in the field of medicine. He is the first Rhodes Scholar in the field of clinical medicine in Hong Kong. And Pei Yong is from mainland China, from a farming village, and he is now the head of a 15-people bioinformatics team. So Dennis is very passionate about training. So both Rosa and myself are two very early students of Dennis. And then we have some students, graduate students, going to the commercial side. And this is Tong Li Yong, our MPhil students. Now he is the general manager and CEO of two of our companies, Exelon and its subsidiary, Sanomics. And Bong and Doris are the laboratory managers of these two companies. And Carmen is the marketing manager of Grail. And this is Nancy. She started PhD at the same time as me, but she looks much younger. And she is now the scientific director of Avalon. And so over the last 4 years, I've mentioned, I gave 143 public education sessions on MPC and associated molecular diagnostics. We recruited and trained over 200 student helpers. They helped in the subject recruitment sessions on the weekends, and we arranged laboratory visits to let them to have a hands-on experience on laboratory testing, and we recognize their contributions on voluntary work. And because of this, the MIT Tech Review in 2015 thinks that liquid biopsy is one of the 10 breakthrough technologies. And fortunately, our group is named one of the key players, one of the three key players in the world in liquid biopsy. And interestingly, the second player is Illumina, but now we have a company merged with this effort. So we play two of the three major roles in this area. So in conclusion, circulating DNA is a treasure trove for molecular diagnostics, and we think that we can develop a tumor marker to detect all types of cancer, and it is a potential for the screening of early cancers before the patients become symptomatic. And I think biotech is a very important development, and it is the best time to do biotech in the history of Hong Kong, and it is an important element for knowledge-based economy. And we are dedicated to train young scientists, and we would like to bring career opportunities to them. Thank you very much for your attention.
Thank you, Professor Chen. Please remain on the stage. May we now invite our facilitator, Professor Lowell, to come forward to host the Q&A section together with Professor Ellen Chen.
Thank you very much for the excellent presentation from Ellen. Ellen, would like to answer the question from the audience. Any questions on their work?
I was rather interested in the biology part of your talk. And I just wonder, because you've got small fragments of DNA circulating, is that because the cells have died and therefore the DNA is fragmented?
Yes.
Yeah.
Please continue, sorry.
Yeah, so I was just kind of curious as to whether or not it might reflect a particular type of cell that's sort of a dying cell. That's why, and then when you do the molecular analysis, then perhaps one might be biased towards seeing the changes that has led to cell death.
Yes, excellent question. So actually, we have put a lot of time in investigating the biology of these DNA fragments. We think that the fragment size is closely resembled to the nucleosome structure. So actually, there are some new studies coming providing further evidence of this, because the nucleosome structures in different tissues are different, because the synchronized of these histone complex are not identical in different tissues. So the DNA are released as a result of apoptosis following the footprint of the original histone packing. So the size of DNA coming from different tissues are slightly different, and the way they are chopped are different in different tissues. So we can find out the footprint by looking at the size and the exact site of cutting in the plasma DNA.
So that's very interesting. So we know that actively transcribed regions of DNA are more open, and silenced regions are more closed. And so that would also be reflected in terms of their nucleosome structure, right?
Exactly.
So are you able to deduce something about the cells from what you see in the blood?
Exactly. So we see that in the plasma DNA, if we look at those actively transcribed genes, the DNA fragments from these regions are shorter because they are more open and they are more susceptible to cutting. So if we look at the regions that are less active in the transcription, then they follow the nucleosome pattern. So we can deduce which genes are active by looking at the size of DNA in the blood plasma.
I have a question. Please correct me if I'm wrong. So that means by detecting the cell-free DNA, now we are able to tell that my body might have cancer, but not sure which type of cancer it is. So by using the DNA methylation method, you might be able to track the DNA origin. But my question is it a prerequisite criteria that CNV occurs in the cancer cell so that we can use the DNA methylation methods to track the DNA of origin? Or if there's other types of mutation like point mutation, insertion deletions, translocations, and or other types of the mutations that leads to cancer, that we can also use the DNA methylation profile to track the DNA of origin, or is there any other methods?
Thank you very much for your question. So I think a question can never be wrong, so only the answer can be wrong. So yes, you are correct. We use the copy number aberrations to identify the source of the abnormality in the method that I illustrate to you. But we are now developing other types of cancers. For example, to look at the mutational patterns and the aberrations patterns, so as to deduce which part of the body is these abnormalities come from. But it is only one. The copy number aberrations is only one illustration of the whole efforts. Because of timing, I have not included everything in the talk.
We may take the last question.
So congratulations on the wonderful work, and as a practicing oncologist, I look forward to a wider application of liquid biopsy. My only question is there an attempt of trying to define what is a clinically meaningful change in the level of a certain mutation in the blood? And is there a move towards like a consensus meeting or consensus guideline amongst scientists and physicians as to guide oncologists like me when to change therapy? Thank you.
Yeah, thank you for the question. So I think for the meaningful levels of of tumor DNA, we need larger scale studies. We are now recruiting tens of thousands of subjects to evaluate what is the meaningful, we want to differentiate signals from noise. So we want to establish the noise level so that we want to differentiate what level of signal can be differentiated from the noise, and we are working on that. Thank you.
Thank you, professors, for insightful sharing. We will have a 10 minutes tea break, and night refreshments will be provided outside the lecture theater. And you may also visit the posters display and demonstration of work at the foyer. Please enjoy.